Good afternoon, everyone. Welcome to the Computational Biology, Data Science, and Epidemiology session. It's my pleasure to co-host this with Dr. Cathy Wu, and to introduce myself, I'm Dr. Michael Whitfield. I'm Chair and Professor of Biomedical Data Science at the Geisel School of Medicine and Director of the Center for Quantitative Biology, COBRE, here at Dartmouth. Cathy, I'll let you introduce yourself.
Thank you, Michael. Hi, everyone. Welcome to the session. So this is Cathy Wu. I'm a professor in the computer science department at the University of Delaware. I also direct the Center for Bioinformatics and Computational Biology, as well as the Data Science Institute at UD. And in terms of the IDEA program, I'm affiliated with both the Delaware INBRE and also the Delaware CTR ACCEL program. So again, welcome everyone.
Great, thank you. So we have a really great lineup of speakers for you today. I think there's a lot of really exciting things going on, and you'll hear about those. We've tried to represent the various IDEA programs across New England broadly. So we have someone from I think every program in each state, really trying to show the breadth of work that's occurring. In terms of housekeeping notes, please keep yourself muted during the presentations. At the end of each presentation, if time allows, we will allow questions to be asked, and those can be either put in the chat, or you can also raise your hand using the hand raise function at the bottom of your screen, depending on what platform you're working from. And then we will unmute you and allow you to ask your question directly to the speaker. So both mechanisms will be available to us. So with that, I want to get us started and introduce our very first speaker. Our first speaker is Kelly Thomas from the University of New Hampshire. Kelly is the director of the New Hampshire INBRE. He's director of the Bioinformatics and Genomics Core at the New Hampshire INBRE, and also director of the Genomics Core at UNH. His talk will be titled "COVID Testing, Tracing, and Genomic Surveillance in a University Community." So Kelly, we'll let you take it away.
Thank you, Michael and Cathy, for inviting me to join you. I'm assuming that you can hear me and see my slides.
We can.
Thank you. So I want to start off this talk basically talking about what I did during the pandemic. Spent most of my time doing this. But the standing up of a testing and tracing and genomic surveillance program at UNH. And I want to start by saying that in the context of our meeting today, that none of this would have been possible without the capacity building that has gone on at UNH and in New Hampshire through our IDEA program mechanisms. So I just want to acknowledge that, first of all. So the goals of our testing program at UNH evolved since, as happened in many places, we did not bring students back in the beginning of COVID after spring break, and spent the summer and the rest of that semester trying to figure out how to have a safe occupancy of campus. And we landed on a very aggressive testing program where our goal was to test every student twice a week. So that's more than 10,000 people twice a week, and all faculty, staff, and contractors. It's another couple thousand once a week. And that this would be tied with really intensive contact tracing and isolation and quarantine of any students who became infected. And in order to do this effectively, that we would have a really minimum turnaround time to maximize the effectiveness of testing in general. And then ultimately to expand our capacity to regional important facilities like long-term care and prisons. This was a very aggressive program and had to be started from scratch. The what we came up with in terms of the sort of not necessarily unique properties, but a combination that ended up being relatively unique were the following. In order to make this doable and reasonably cost effective, we started with a pooling strategy. And so we have a surveillance mode where we pool 4 individuals together for every test. One of the more interesting and innovative approaches was, in order to get all of these samples and tests done by all of these students, was a self-swabbing approach. So students were provided with swabs and dates and times to submit their samples. One of the interesting things that this turns out is, unlike other testing programs which tend to have reasons that they're testing people that have to do with either symptoms or contacts, ours is a relatively unbiased testing. Basically, everyone on campus had to test and was tested. As a result, one of the things is that, and because of the frequency, most of our positive tests are probably very early in the infection process, which has some interesting analytical things downstream, which we're interested in. In order to make this work, one of the things that was very important, because we had the capacity through our INBRE and COBRE programs, was the ability to develop a computational system, a LIMS system, that would actually guide this process, and really was the fundamental glue that brought everything together and made it possible. And the same day, basically, testing, contact tracing, and isolation, that I'll tell you how that was possible. And ultimately saving all of our positive specimens throughout that period of time so that we could conduct genomic surveillance subsequent to that. So designing this involved and implementing it involved a very diverse advisory committee, which I have in my acknowledgement slide. But basically, everyone, scientists, primarily myself and Rick Cody, our COBRE PI here, medical staff, and the administration of the university all had to be, rowing in the same direction in order to make this happen. Of course, it had to be equipped. We had none of the equipment, and as everybody understands, all of that equipment is useless if the supply chains aren't there. So there was a lot of time spent on the phone and emails, making sure that we could get everything we needed. We ended up actually setting up 2 testing laboratories. One of the other ones is part of our INBRE program, and that's at UNH Manchester. They set up a separate, sort of completely separate supply chain system with doing LAMP-based tests. And we had to set up a new CLIA certification because we have not had CLIA on campus for a number of years. So just to go over, this is a one slide of how this testing system works. And that is, we start with these swabs that you're all aware of, coming in by the thousands. Those have to be reformatted into 96 well plates. The plates have to be combined in a pool, and then the RNA extracted, and testing of those pool tests. And when we had the pool test, we would immediately, what we call demux, we would unpool them and then look at identify the individual that from this surveillance step. And one of the important things was that was so accurate, even though it was not the CLIA test that was the ultimate test, that was so accurate that we could begin the contact tracing and isolation of those individuals at that time. And then that was followed up with a medically administered test, so not a self-swab, and a CLIA-approved PCR test, all in a very short time period. So to cut to the chase, this is what our epi curve for the testing looked like. You can see at the beginning of the fall semester, we were processing large numbers of samples and had very few positive tests. So life was great at that point. And then we went through a series of events on campus, which were luckily short-lived. And I think in part that has to do with the ability to test and isolate people very quickly. I won't belabor this point. You know, the testing, of course, dropped during the holidays and then came back. And we'll talk about more specifically about what some of these more challenging periods of time had to do. But you can see the total numbers of tests. So we've run over 700,000 tests combined between the two facilities. And as I said, saved all of these samples for subsequent sequencing. The key properties that are important to point out here are that because of the way that we designed this, we were able to eliminate a lot of the problems. So basically every sample is tested 3 times, twice on the original sample, which allowed us to get rid of the pervasive inconclusives, which everybody has at about a 1% level. So ours went down to, 0.1%. And ultimately, because of the way that this ended up being designed, I believe that our false positive rates, which are basically impossible to calculate, are extremely low. And so that gave us a really strong benefit throughout the semester. Ultimately, the turnaround times were very short. Our tests, basically on an average day, tests received that day were processed through the first surveillance, and that night or early in the following morning, contact tracing began, and the STAT or CLIA test was conducted in the following day. So basically a 24-hour turnaround time. We throughout this process, those of you that know me know that RT-PCR is not my love, but sequencing is. And so I always wanted to be able to conduct sequencing. And this is a picture, so I can do a little name dropping. The person with her back to us in the orange is Deborah Burks. She visited us a few times, and her comment was always the same to me as, why aren't you sequencing? And so the university became convinced and invested in the sequencing facility, and we also have a subsequent supplement grant to conduct that sequencing. And the sequencing is important. I think most of us on this call, or on this Zoom, understand that, of course, the virus changes every time it replicates, and some of those changes can cause these variants of concern. And what we need to do is to understand what we're dealing with in terms of all of these variants. So we want to understand issues like, are specific genotypes or variants, do they have increased infectivity? Do they have different symptomatic severity or reinfection potential? Or is there specific strains that are able to undergo vaccination breakthrough? Are viral loads different between strains? And what are there differences in the outcomes of testing and tracing? So the sequencing protocol that we used is a very common one. It's basically an amplicon-based system. We amplify the virus in 98 different overlapping amplicons in 2 batches. And there's some human RNA control that's thrown in there. And then those are mapped to the reference genome, and all of the variable positions are identified to produce a new version for every positive sample. And then those are compared to the database of known variant sequences, and so we can identify those and conduct phylogenetic analysis and identify the different lineages that appear in our samples. So this is our epi curve that now has, on a weekly basis, all of the different variants. And so a couple of features that you can see in here is that things change over time. We have, a diversity of lineages on campus all the time. Not so much anymore. But we went through a couple of periods. Surprisingly enough, our first sort of event of note was Halloween. I didn't realize it was that important a celebration time, but the real one that really kicked us in the rear was the Super Bowl, which you can see. Of importance to all of us was the emergence of some of the more notable variants of concern, in particular the UK variant and the P1 variant, the Brazilian variant, which did arrive on campus, while they took over the proportion of the positive cases, so basically everything became that, and before the summer break, we were able to keep that in check. And at that same time, we started the vaccination clinics on campus, which has also helped immensely. Just another little taste in the last little slide or so here is, for things, as an example for reinfections, while the number of reinfection cases we have so far is actually very small, some of the questions that we have, for example, are the titers, which are reflected in our case imperfectly by CT values, do they increase in reinfection, or do the titers decrease and the CT values increase? And so while it's not significant, you can see a trend that's in the right direction. But more importantly, what we were able to find is that reinfection strains are always different. So these are in fact reinfections. And the reinfection strains, the ones that reinfect, are just a representation of, at that week, what were the strains that were on campus. The other thing that this has allowed us to do is to combine our contact tracing information with the lineage information now. And so what you can see from this pretty picture with all of these connections, every one of these are positive cases, and the contact information with linking them. And what you can see, and then they're colored by the lineages. So what you can see overall is, in the vast majority of cases, all of these contacts that were known from the wonderful contact tracing that went on were from the same lineages. And then we can address the ones that don't appear to be that way, and understand how each of the lineages behaves differently in this context. To end this, I want to acknowledge there's a whole bunch of people that it took to do this, even though it was highly automated. And I want to call out Tony Westbrook, who is part of our INBRE program, who really designed the LIMS systems, and then subsequently Molly Kephart, who was also an INBRE alum and part of the amazing techs that stood up this lab, and is now heading up our sequencing surveillance program. And there isn't enough time to talk about everyone here that are just, these are amazing people to work with, and represent a tremendous capacity building for us, and I think for the state. And finally, just a shout out to the UNH task force that built this. It really was represented by a bunch of people that were able to make some hard decisions. And I've highlighted Rick here, who was my partner in the development of the scientific side of this. And with that, I'll end with acknowledging CIBRE, and which is our COBRE, and the New Hampshire INBRE program. And I should note that specifically the surveillance sequencing part of this is now supported by a supplement to the CIBRE grant. Thank you.
Thanks, Kelly, for a really nice talk. It's now open for questions. And so you can either raise your hand or add questions to the chat. And while people are getting their thoughts together, I'll ask a question to start. So obviously we're now in an uptick of new cases and spiking vaccine breakthroughs. Can you comment on your the cases that you're seeing, case loads, and even numbers of Delta variant that are now showing up in current sequencing?
Yes. So we are, as of Monday, we are back to testing larger numbers. We went down to hundreds. If you saw the y-axis on some of those, the hundreds was really boring. Now we're back up to 1,000 or so a day. Most of the summer we had nothing, and then over the last few weeks we have had a few positives, still very small numbers. They are all, as far as the sequencing has gone, they are all Delta. variants, which is not surprising to anyone. And it's my understanding that they are virtually all, although it's a very small number of breakthroughs. And that I should make it clear that virtually everyone that we test is vaccinated. So there weren't going to be any other ones. I hope that answers your question.
Yeah, thank you. Other questions? Feel free to unmute yourself and ask if you'd like.
Kelly, spectacular talk. I mean, that's the scope of what you guys carried off is amazing. I'm curious, did you have to add security protocols dealing with human data as opposed to all the other things? Did you have the capacity? Is that something you had to really blow up?
Oh, yeah, you know, and Joel, you know me well enough that my nematology background did not prepare me for the clinical aspects of this at all. But yes, so yes, the answer is yes. That part of it, that connection with the health records part, was one of the more expensive parts. So yes, the answer is yes. Just that whole process, even the CLIA process for the lab, was something that I had not gone through before. But certainly in terms of, and we still deal with this in terms of our sequencing, surveillance sequencing now, because there's lots of privacy issues, and so we have to make sure that we are respecting the privacy of all of our students and faculty and staff as well. Lots of time with IRB, and thankfully my colleague Rick Cody is really good at stuff like that.
Any other questions? All right, thank you, Kelly. That was really exciting to see the things you're doing at UNH. And you know, as we're at Dartmouth, we're looking forward to working with you on these types of things going forward. So
yes, thank you.
Sounds great. And that's a wonderful segue to the next talk by Dr. Joe Graber from MDL Biological Lab. So Joe is a senior staff scientist and director of computational biology at MDL Biological Laboratory. He also co-directs the main INBRE bioinformatics core, as well as direct the NH-COBRE functional genomics core. So certainly wear multiple hats. And this is a wonderful continuation of what was covered by Kelly. His talk is best laid plans reviewing the NH-COBRE computational biology core response to the pandemic.
All right, well, I would also like to thank the organizers for the invitation to talk. This is going to be a very different scope than what you just saw. So rough outline of what rough outline, if I can get, there we go. I want to talk just briefly about what our role is as the core and how we were affected. A little bit of how we went about changing what we were doing, how we adapted. And then talk about how really what I'm trying to do with this as we move forward is learn from it and change the way we're doing things in a way that takes advantage of what we learned and what we had to do differently over the course of the last year, treat it as an opportunity as much as a negative. Again, talking about the scope, and this is my entire team right here, myself and the other two people. And as Cathy said in the introduction, we're wearing a lot of hats, and so we really have to try to be efficient with our work. And so, you know, we talk all the time about rigor and reproducibility. We talk about not wasting efforts. And so we really, you know, as I said, this is a small team. And so when the pandemic hit last year, we had to look at what we were doing. We had to look at what the laboratory was doing, especially in the context of the INBRE, which is where I'm going to spend most of my efforts today as we talk about it, in terms of keeping things going, in terms of keeping things moving, and hopefully being successful successful in making this go. So here is what it is that we do in the INBRE. This is the written specific aims for the INBRE Bioinformatics Core. As Cathy said, I co-direct this with Ben King at the University of Maine. But we have these two goals. We are working to help our research teams at our partner institutions. Our partner institutions are primarily undergraduate institutions. We have faculty there who are doing wonderful work, but they are largely tied up in education. And so it's our job to help them get the computational resources, to get the knowledge and information they need to make things go. We also, as the second aim here works, and this is where a lot of my effort, what we're going to talk about, what we did for the last year, is in helping with the educational programs, helping to try to reinforce the application of statistics computational efforts in those problems. And as it says here, I really view our role, especially my role and the people working with me, as being a conduit. It's our job to help these people who have heavy course loads of what they're teaching get access and the knowledge to use external compute, use the tools and materials that are available, because they don't have the time to do this themselves. We do this in the context of a set of guidelines, of goals. They're things that we're trying to reach. We're more closer there than in some areas than we are in others. But primarily at the top, we start with the basic idea that everything is being run through command line protocols, workflows that are run at the command line through scripted control. The resources, as I'm sure all of you know, carrying out this work requires an enormous number of external resources in terms of programs, in terms of libraries, in terms of sequence databases. We maintain and manage those ourselves within the core. That keeps a consistency, that keeps predictability of where they are, it keeps the ability to keep track of which ones have been used. We wrap our scripts in order to be modular in terms of reworking workflows. And in the ideal, we want the people that we're working with, whether there are the courses, the students in the courses that we're helping to teach, or the research partners off at the undergraduate institutions, that if we've done our job properly, they're mostly working with configuration files that can be handed off to a script that will carry out a designed, planned workflow that will get them to the answers that they want. And as it says, we're really always trying to revise and look at these things in terms of enhancing the ease of use of these, so that the possibility of mistakes or misunderstandings are reduced. So with that framework in mind of what we're trying to do, how did last year change things? Well, I think most of you know MD-IBL is the host institution for the main INBRE. This is a map of our various partners within there. And in most years, this is what it looks like. We have a whole lot of people coming in, and this spreads across multiple aspects. We have conferences and workshops that we host. We have a very rich summer program where we have high school and undergraduate students who come in for summer internships. And we have courses with each of these that we share with each of these partner institutions where typically they're hosted here on site. It's a joint venture of faculty here on site at MD-IBL along with faculty from the partner institutions. Of course, last year, like every place else, we ended up shutting down. We ended up with limited visitors. We were working remotely through part of this. So this broke the model of what we have been doing historically. So we had to go to a computational method. We had to go to virtual learning like everybody else did. And we had to make a decision, how were we going to carry out these courses? A standard way that a course, one of the short courses with our undergraduate institutions, would be that they would bring a group of people here for a week. And they would work intensely in the lab, and we would give some auxiliary lectures to help with the computational part. However, this was just not a possibility. So we had to make some decisions. How were we going to carry these out? Were we going to try to carry them out at all? And if we were going to carry them out in a computational nature, what was the way that we would do it? There are lots of choices for how you can do this. You can use external servers like Galaxy. You can use dedicated resources on local compute. There are any number of doing it. So given what I've already told you, we made the decision to teach from the command line. This was not a trivial choice, but we decided to do it. And our primary rationale for that is that, just as I said at the beginning, our belief, my belief, is that scripted command line workflows, if they're well designed, if they're built to self-document, to generate the log files you need, they're the most direct path to getting work done reproducibly and robustly. That's just what we believe. As Dave Barry has said many times, it seemed like a good idea at the time. So what is it that makes this work difficult? Well, for the students, here's the starting place. It takes 3 sets of knowledge. I am going to assume that some of you guys have experienced this before. In order to make your computational analysis, in order to make your bioinformatics analysis go, you first have to really understand your biological system in your question. You have to understand the concepts behind what does it mean to align sequences? What does it mean to index sequences? What does it mean to pre-process these things? But finally, and possibly the most frustratingly for many people, the third area of knowledge that it takes is how do you maintain, how do you execute, how do you install these things in a way on your local computer system? And that's the one where ideally we are doing the work with our partners and we're helping them to make this last part go. What makes that part of the job difficult in what we are trying to do? And that is, as it says here, this is my standard slide on this. This field has grown very rapidly, competitively, and much of the work that we rely upon, many of the programs are not the primary end goal. They're a byproduct of the research. They're written by grad students and postdocs. My favorite phrase that I get from people is, "It will just work." Sometimes it does. A lot of the times it doesn't. And so it takes a lot of work to keep these things going. They can have conflicting resources. There are lots of tools, lots of approaches to trying to make this easier. But they're all works in progress at this point. We also have auxiliary data and a special that comes in many different forms, taken by different means by different programs, and trying to pipe this all into a standardized workflow can be quite a challenge. And the last one that is a little bit special to us, not unique, but a little unusual, is because we're working with so many partners. We're trying to make these systems, we're trying to make our tools such that they can be transportable across multiple systems, we work with the compute cluster people at Bowdoin, at Colby. We're working with the University of Maine Computation Group. We're spinning things out on Amazon ourselves. And so that creates a whole lot of extra challenges that we had to deal with in making this go. So I want to talk about just briefly three examples of what we did to keep our programs alive over the last year. Again, as I say, this is a much smaller scale than we talked about than what we heard Kelly talk about. But this was our focus in trying to keep things alive and going. So these are the 3 programs that we managed to keep alive in some way or another. So the first one is our non-model organism transcriptome workshop. This is something we're coming in for our 3rd year of this coming fall. Here's how it changed last year. The previous year, we had a 1-day event in-house. We invited people in, they came in, and we spent an intensive Saturday working through various aspects of transcriptome assembly, analysis, annotation, and such. This was not going to be a possibility this year. So we transitioned, we decided to make this a virtual experience. We distributed the lectures and the meetings over 4 days instead of 1 day. We pre-recorded lectures for asynchronous viewing, recorded them so that they could be viewed as needed. We made machines available for all of our students on Amazon Web Services, and provided markdown documents with materials and teaching tools for them to work with. And then finally, we met with them by video at least twice every day and recorded those and kept them available for others. We had 16 registrants as opposed to about 20 the year before, so we feel like it was fairly successful. In most of the meetings, there were usually about 10 people showing up, or 5, as it says, 5 to 8. We had 10 people who were truly actively involved in making this go. And it's worth looking at and saying, well, okay, this was a different venue, a different mechanism by teaching this, but what did we gain from it? Well, as it says here, we got the ability to work on more realistic problems because we could spread the compute time across multiple days. We had really, in my view, we had a lot more time for our participants. Doing it in one intensive day, it's hard for the students to really keep up with it and ask questions. And by going across multiple days, I really felt like we had students come back with better questions, with more understanding of what we've been trying to talk about. And we also, of course, anytime you run a course like this live, you run into issues. And so we had the time to catch up with those. Here is one of the meetings that we had within there, smiling faces. So I'm happy to say that that went well. The next thing that I want to talk about is the summer program. Trying to get the chat window to wake up here because I see there's the chat. Okay. Okay, never mind. So the summer program, the first and foremost thing that we really wanted to do was to see if we could make sure that this happened. My understanding is that nationwide, about 75% of these programs like this were not even held. They simply were canceled. So we went ahead, we decided that it was worth continuing to make this happen. We ended up having a summer research internship for 23 different students, 19 undergraduates, 4 high school. You can see the distribution on here. This represented 15 different institutions across the country. We recruited mentors from different institutions within our INBRE network. We had 7 different institutions who gave up 16 mentors who all agreed to work with us in this unusual format. They had to be willing to come up with a project that could be done in a computational format. And then at the end of the summer, we held a symposium on that. Our role in making this go was we live streamed several video lectures to the students to help them. We did twice weekly. We held open office hours by Zoom, or we also were available by direct email. This was my team and myself, again, being available to help with students with the projects that they had going on. We did direct mentorship for 4 students, 2 high school students and 2 undergraduate students, working with data sets from Jim Kaufman's lab, zebrafish expression data. This is actually work that's ongoing and nearing a state of being able to be written up for publication. And finally, we also built a series of tutorial documents, what we call mini workshops, that we made available for all the students. And this is something that we are now trying to package and get into a better format that we can share beyond just our summer student program. And again, here's another successful picture of what happened within that. The final thing that I want to talk about in terms of these programs is our short courses. As I said, these are usually held in person. We did, by the end of this school year, hold one in person here. College of the Atlantic, of course, is only about 2 or 3 miles away from us, so it didn't involve expanding any bubbles too much. Other than that, we took on 2 new courses that we had not been responsible for within the core before. We worked with faculty at Southern Maine Community College with a goal, also with James Godwin here locally at MDIBL, looking at RNA sequencing analysis of axolotl regeneration samples. And this was a course where we took on what had been the same time format. We did a one week long every day, doing lectures by video, doing meetings by video, and again, giving the students access to compute resources off on Amazon. We also, in a very different format, worked with Hadley Horch at Bowdoin College, again, building upon the information and the materials from the Minota workshop. We worked on continuing to help with the refinement and the analysis of the building of the transcriptome assembly for the cricket, which Dr. Horch works with there. And in that case, we sort of had to fit into what the constraints were that had been placed on the course by Bowden, in that they were only allowing half of the students to be in the laboratory at a time. And so we worked out this interleave schedule where half of the students were in a computational meeting one week and the other half were in the laboratory, and then we flipped it across that. And we did that through this whole semester. This last year. And here is again an image and a link to a very nice write-up that we got within the Southern Maine Community College Public Relations Department. So looking back on that, what went well? The primary thing that I can happily say is that we managed to maintain our efforts. We carried out and met the goal of what we wanted to do in terms of at least these programs were held, they carried forward. The student feedback for the most part was very positive. The work at the command line, which while it's very difficult and challenging, something they haven't seen before, it gave them new skills, and they were quite positive in what they felt like they learned and were able to do. Our tools and approaches were expanded and used in new systems. And importantly, and this is going to come in the next slide, we got feedback. We got vetting and feedback, and we're revising and trying to respond to that and change what we're doing. What didn't go as well? The primary one is right here. We frequently underestimated just how much of a challenge it was to take these students who were not used to working at the command line and bring them up to speed. And as it says, when trying to generate these tools, when trying to generate the mechanisms, we have to minimize the chances for things to make mistakes. Our configuration was primarily designed for our work in a research endeavor, and expanding that to an educational system, it was challenging, and it didn't go quite, those are not the same thing, and we're trying to learn from that. And so, of course, accordingly, projected milestones and timing slipped. We didn't get as much done in these courses as we hoped we would. So final thoughts to wrap up on the whole, I'm pleased. I feel like in terms of the INBRE program, we fulfilled our core mission. We got through it. We received a good testing from which we can learn, adapt, and iterate. And as I sort of started in the early parts of this talk, I want to learn from this. I want to take this forward. And so accordingly, probably the biggest change we're going to have is with the Minota workshop this coming fall. We're going to do a fully hybrid approach. We're going to lead into it with 4 or 5 days of virtual distant connection, and then host an insight final meeting of that to discuss it in a way of trying to combine and build the best from both of these. And to end again with another quote, I'm hoping that we can continue to develop our set of tools and come up with things that will continue to solve the problems that we have and work better for our partners here. And acknowledgements, of course, all the people that I worked with, the people at Southern Maine Community College, Bowden and Colby have been great. And so, thank you.
Thank you. Thank you so much, Joe, for sharing those very creative training educational programs and initiatives that you developed and share with the lessons learned. I'm sure it really resonates with our colleagues here. So are there comments, questions for Joel?
Hi, Joel. Great talk. I was just wondering, the transcriptome data sets that you had people work on, did you have everybody work on the same data set, or did I understand that everybody brought their own data set and that was how you had them move forward?
It was a mix, and especially in the Minota workshop, we've always encouraged people to bring their own data if they have it. But we also pull data externally, and we have some modest sized data sets, especially back from the days when we were trying to do everything in one day. We had some small data sets that allowed for rapid analysis of that. But this is an evolving thing. It's always a difficult question to come up with the best way of setting these up. But we had, in terms of the Bowden course working with the Cricket, they were all working with common data set.
Thanks.
Thank you.
I had a question as well, if I could go next. Yeah, I was curious about the hybrid model that you mentioned and sort of how you envision that going forward. Because I think we did some similar workshops at Dartmouth last year, and I think we got a lot of external people that wouldn't necessarily have otherwise attended our workshops, using the online format. And I was wondering, I'm thinking for your hybrid model, having people do part of it online and then be partly in person. Are you also thinking about a hybrid model where you have some people attending fully remote or some people attending, in part? And what's your thoughts on that, I guess?
That's a great question. And at this point, my plan is to leave it as open. I'm hopeful that people will be able to come in. You saw the map of Maine. Maine's a big place. Asking someone to come here, for most of our partners, coming back and forth on the same day wouldn't work. But one day is not such a big deal. My plan at this point is to go largely with what we did last year in terms of the lead up, with asynchronous recorded lectures, with material, with help sessions, 2 or 3 hours of Zoom connection, to do that in a week leading up to that final Saturday. I'm hoping that people will be able to come in for it, but I'm leaving open the option that we're going to be streaming it as well. Because, we did last year. We had, I think, 1 or 2 people from Ohio who were among those who connected in with us. And that's a lot further. And as you said, it opens up opportunities for people to join with you who wouldn't be able to make the trip as easily. So it's going to be a work in progress. Go ahead. You wanted to say something else?
I was just, obviously there will be challenges with trying to do like an in-person and a hybrid, like in-person and remote at the same time. And I guess I haven't thought of a good solution to what those would be, but I'd love to hear if anyone comes up with one. Yeah,
if anybody else has it, jump in. But I mean, I'm going to be working with our IT people and, trying to make sure. The biggest challenge that I find in this mixture of video and in person is having enough of a visual of your remote people so that you actually feel like you're interacting with them. We fortunately have some real nice monitors around here now, where I'm hoping that if we can get that in the right place, where we actually really see, it's almost, it's body language and such.
Sounds great, Joe. So I think we should move on to the next speaker. But definitely, this is a topic that potentially, if we still have time at the end of the session, we can come back, because again, I think there are a lot of lessons learned and ideas to be shared among the participants here.
Yep, I agree. And I would also encourage you to continue any discussions in the chat if you'd like to. You can ask questions and speakers can answer them after their talks are done. So it's my great pleasure to introduce our next speaker, Dr. Erica Moen, who is an assistant professor in the Department of Biomedical Data Science here at Dartmouth College in the Geisel School of Medicine. Erica is also a project leader on the Center for Molecular Epidemiology, COBRE, here at Dartmouth. And today she's going to tell us about her work on diffusion of genomic testing in breast cancer care. So Erica, welcome. We'll turn it over to you.
Great, thanks, Mike. All right. So thank you to the organizers for the invitation. I'm really thrilled to present my work to this group. This is the work that was funded through the COBRE, and I studied the diffusion of genomic testing in breast cancer. And I think, just to continue on some broad theme from the previous talks, which were really interesting, it's the idea of how we translate scientific research and science policy to what's actually going on out here in the real world, and what's working and how can we improve. So every year in the United States, approximately 100,000 women are newly diagnosed with early stage breast cancer. And one of the important clinical decisions these patients have to make with their providers is whether they want to include adjuvant chemotherapy in addition to hormone therapy. And this decision is not taken lightly, because chemotherapy is a very toxic treatment with serious adverse effects. So the goal is to spare patients these toxicities if they're unlikely to have any survival benefit from chemotherapy. And so over the past couple of decades, we've been able to capitalize on the advancements in genomics to identify breast cancer patients who are at low risk of disease recurrence and therefore unlikely to benefit from chemotherapy. One of the most well-known genomic tests in breast cancer and maybe cancer in general is called Oncotype DX. And this test measures the expression of 21 genes from the tumor. And depending on the expression levels of these genes, it can predict whether a patient is at low or high risk of disease recurrence. So the way this test works is once the decision has been made to prescribe Oncotype DX, some of the tumor mRNA is sent out and the test is run, and it basically returns a numeric value called a recurrence score between 0 and 100, 0 being on the low end and 100 being on the high end. And patients classified as low risk have a score of less than 18, and they are recommended to receive only hormone therapy and not adjuvant chemotherapy. Patients in the intermediate risk range with a score of between 18 and 30 originally were recommended hormone therapy plus adjuvant chemotherapy, but subsequent large randomized clinical trials of this intermediate risk population have actually shown that some of these patients also don't receive a survival benefit from receiving adjuvant chemotherapy and are therefore recommended to only have hormone therapy as their treatment. And then patients at high risk of disease recurrence with a score of greater than 30 are recommended hormone therapy plus adjuvant chemotherapy. So when thinking about utilization of Oncotype DX, there are several patient characteristics and clinical characteristics that are important to consider when deciding whether or not Oncotype DX is appropriate for a patient. So a patient's age might come into play. Older patients are less likely to tolerate chemotherapy very well. So if the physician is unlikely to prescribe chemotherapy to a patient no matter what, because they're of an older age, that might indicate they're not a good candidate for Oncotype DX. Of course, breast cancer stage is important, their lymph node status, and their hormone receptor status. However, studies have shown that physician characteristics are also associated with utilization of Oncotype DX. And some of these characteristics suggest that there might be unwarranted variation in utilization of Oncotype DX depending on the types of physicians that are providing care for the patients. So for instance, physician familiarity with genomics has been associated with their utilization of genomic tests in their clinical practice. This is pretty intuitive. If a physician just doesn't feel familiar or feel comfortable with genomics, their ability to communicate genomic tests to the patient, they're going to be less likely to use the test in clinical practice. Clinical experience has also been associated with utilization of genomic tests like Oncotype DX. And by clinical experience, I mean the number of years since the physician has graduated from medical school. And studies have shown that more recent graduates are more likely to incorporate genomic tests like Oncotype DX into their clinical practice. And this is likely because they received formal genomics training in medical school compared to physicians who have been practicing for decades, long before genomics were part of clinical care, and who might be more set in their ways. And also local practice norms have been associated with Oncotype DX utilization. So has the practice as a whole decided to incorporate Oncotype DX into their clinical workflow? Are a physician's peers using Oncotype DX and therefore increasing their awareness of the test? So with my study, I aim to examine whether exposure to early adopters of Oncotype DX increased likelihood of a medical oncologist utilizing Oncotype DX. The data source for my study was nationwide Medicare claims data from the years 2007 to 2014. So Oncotype DX was developed by Genomic Health in 2004, and then CMS covered Oncotype DX starting in 2006. So for our entire study period, Oncotype DX was covered for all Medicare beneficiaries who were eligible for the test. And what you can see here on the y-axis is the year of our data, and then the x-axis is the year of the data. The y-axis is the number of providers who prescribed Oncotype DX. And this blue line here is when they are a first-time prescriber. And we have a couple events here of note. So the American Society of Clinical Oncology incorporated Oncotype DX into their evidence-based clinical guidelines for early-stage breast cancer patients towards the end of 2007. And then NCCN, another national society, followed suit right at the start of 2008. And what you can see is right around the time of inclusion in the ACCEL guidelines, there's a really substantial increase in the number of providers who adopted Oncotype DX for the first time. And then right after the NCCN guideline, there's also a little bump in first-time prescribers, which we think might be due to the inclusion in the NCCN guidelines. So for our study timeframe, we focused on the years 2008 to the end of 2013. And this is because for these claims-based algorithms to identify breast cancer patients, we required a look back year, which was 2007, and a look forward year, which was 2014. So we considered years 2008 to 2010 to be the early time period. So if a physician was a first prescriber in this time period, they were considered an early adopter. And then the years 2011 to 2013 was our late time period. So if we observed a physician in the early time period, but they didn't prescribe for the first time until the late time period, we called them a late adopter. In our data, about 70% of the Oncotype's prescribers were medical oncologists, and about 20% of Oncotype DX prescribers were surgeons. And this is pretty consistent with what has been observed with in other studies. This is the first time studies have looked at nationwide Medicare data, but it's very similar to what was observed in SEER Medicare data. And the clinical workflow for breast cancer is typically that you see your surgeon, the surgeon removes the tumor, and then if the surgeon thinks that, or if the surgeon, if the clinical characteristics of the tumor indicate that you need adjuvant therapy, the surgeon would then refer you to a medical oncologist. So at which point it's typically the medical oncologist that would then decide whether or not the patient should be tested with Oncotype DX. So the work that I'll be showing was recently published in Cancer Medicine earlier this year. And what we were really interested in was the impact of peer influence on Oncotype DX adoption. And in order to study peer influence, we assembled networks of physicians. And networks are, there's been a lot of research in the social networks literature on the importance of network connections in both diffusion of new innovations and the role of peer influence. So if what we did was we assembled these physician networks. Here's an example here where each of these circles or nodes would represent a physician, and lines between the two nodes indicate that there's a relationship between these physicians. And in our study, we considered two different types of relationships. We built relationships based on sharing common breast cancer patients, in which two physicians are connected if they shared for if they cared for the same breast cancer patient. But then we also thought, well, there are situations such as two medical oncologists being part of the same practice, but not sharing patients, and they would still be important to the, to the diffusion of innovations among those physicians within their practice. So we attempted to identify these types of relationships, calling them co-located physicians if they practiced in the same zip code, which is the most granular we could get with the Medicare claims. And then we also characterized edges as being both sharing patients and co-located if we observed both of those types of relationships between the pair of physicians. So we assembled a network in the early time period, and we identified the early adopters of Oncotype DX. And then we assembled a network in the late time period, and we were essentially asking whether physicians who were connected to early adopters in the early time period were more likely to then adopt Oncotype DX in the late time period compared to physicians who had no connections to early adopters in the early time period. So the first model we ran was a physician level model where the outcome was utilization of Oncotype DX in the late time period. And this was among physicians who had not prescribed Oncotype DX in the early time period, but who we observed to be practicing in that time period. And it was a multi-level model where physicians were clustered by hospital referral region. We found that patient volume was a strong predictor of adoption of Oncotype DX. And then we considered relationships to two types of early adopters, relationships to medical oncologist early adopters and relationships to surgeon early adopters. And what we found was that co-location with early adopting medical oncologists was positively associated with Oncotype DX adoption, and connection to early adopting surgeons through co-location and patient sharing was associated with increased adoption of Oncotype DX. We then ran a patient-level model, a multi-level logistic regression, where the outcome was the patient receiving Oncotype DX in the late time period. And we included patient and medical oncologist level predictors in the model. We found that older patients were less likely to receive Oncotype DX, and this is consistent with previous literature and likely for the reason I stated before, as older patients are less likely to be prescribed chemotherapy regardless. So if the physician would be unlikely to prescribe chemotherapy no matter what, it wouldn't be cost effective or timely to use Oncotype DX. Black patients were significantly less likely to receive the test compared to white patients. And this has also been seen previously in the literature in SEER Medicare data sets, and I think in National Cancer Database data sets as well. In terms of medical oncologist predictors, we found that female medical oncologists were slightly more likely to prescribe the test compared to, or patients treated by female medical oncologists were slightly more likely to be prescribed the test than those treated by male medical oncologists. And then for the network effects, if the patient's medical oncologist had been connected to an early adopting surgeon through co-location or both co-location and patient sharing, they were slightly more likely to receive the test. And then we thought it would be interesting to visualize geographic variation in Oncotype DX testing. The denominator is patients who are likely eligible for Oncotype DX testing. We used a validated claims-based algorithm to detect eligible patients, and then the numerator was the number of patients who received the test. And this is a map of the United States bounded by hospital referral region, which represent regional healthcare delivery markets. And as you can see, there's a lot of geographic variation in the percent of eligible patients who receive Oncotype DX, ranging from 5% of eligible patients to over 30% of eligible patients. And so there's clearly a need to understand what's contributing to this geographic variation to ensure that no matter where a patient lives, there is equitable access to genomic testing in breast cancer care when it's appropriate. One of the difficult things is we don't really know what the optimal percentage of patients who are eligible to receive Oncotype DX should receive Oncotype DX. No one's thinking that 100% of eligible patients should receive the test because there's a lot of other clinical parameters that go into these decisions. But the fact that there is this much geographic variation does indicate that there are some sort of clinically based reasoning for prescribing the test. And since in our study, we saw this really important role in early adopting surgeons in terms of the diffusion of Oncotype DX, we also mapped for all of the hospital referral regions the percentage of prescribers who were surgeons. This is sort of an interesting area of research in the recent years as well, because since surgeons are earlier in the clinical workflow, there have been some studies that have suggested that health systems should recommend that surgeons be the one to prescribe the test because it will minimize the delay in beginning adjuvant treatment that might occur if the medical oncologist is the one who orders the test because the medical oncologist sees the patient after the surgeon. So if a surgeon identifies someone who is a good candidate for Oncotype DX, it might be better for them to just order the test themselves to reduce the delay that might occur as the physicians are waiting for the test results in order to make a treatment decision. And we do see, though, substantial variation in the proportion of surgeon prescribers across the country. And we saw that there was a particularly strong correlation between the percent surgeon prescribers and the percent of patients who are eligible who received the test, especially in these less populated areas, which may suggest that in less populated areas, it's beneficial for surgeons to be the ones prescribing the test, and that might be resulting in higher quality care for these patients. So the strengths of this study are that it was the first time adoption of Oncotype DX and the peer influence of Oncotype DX has been studied using a nationwide cohort. We were able to identify relationships based on patient sharing ties and through co-location. And we also considered interdisciplinary diffusion. So rather than just diffusion based on relationships among medical oncologists, we also considered ties to early adopting surgeons. The limitations are that this was a Medicare beneficiary cohort, so we didn't have data on patients younger than 65 years of age. We can't account for other sources of exposure to Oncotype DX. We used a claims-based algorithm to determine Oncotype DX eligibility because we didn't have access to registry data, which would have more definitive information on hormone status and stage. And we, of course, can't account for patient preference, and patients, especially breast cancer patients, are known to advocate for themselves and to be, so there might have been a role for patients advocating for Oncotype DX testing for themselves, which we weren't able to observe in the claims data. So I would like to acknowledge the people in my group who did a lot of the heavy lifting in these analyses. Margaret, the COBRE PI, my COBRE mentors, my other collaborators, and my funding. And I don't know if there's time, I'm happy to take questions or respond in the chat.
Great. Thanks for a really nice talk, Erica. Open for questions if anybody would has anything they would like to ask. And actually, I'll start by just, I was curious, looking at your table, that being at a teaching hospital didn't seem to matter, if I read the table correctly. Was there any particular kind of setting other than these networks where you have early adopters that they were more likely to prescribe these tests?
Yeah, we were surprised we didn't see a teaching hospital effect either. Part of the limitation in claims is it's actually sometimes difficult to determine the facility at which outpatient care is occurring. So we have zip code, and then in some claims we can get the facility, but physicians practice in multiple facilities. And so that's actually a little bit trickier to determine when you just have claims data. But I agree, it would have been, we started to pursue a line of inquiry where maybe the early adopters are all these like NCI cancer centers or these hubs that then it diffuses out. And we just didn't really see that in the data.
Thanks. Other questions?
I see some in the chat and I can just respond in the chat in order to keep on time.
Sorry. Yep.
Okay.
So Cathy, yes, I'll hand it over to you.
Yeah. Okay, thanks. Well, so we are delighted to welcome our next speaker, Dr. Laurent Huber Dufresne. So he is associate professor of computer science at the University of Vermont Complex Systems Center, and also project leader of the Translational Global Infectious Disease Research Center. And you can see his title on the cover slide there. So take it, Laurent.
Thank you. Can you see if I change my slide? I get a message that sharing is paused. Are the slides moving for you?
This is on your cover right now.
Okay, let me, and resume share is not working. Let me just try again. Still not moving for you?
Right now we are looking at the messages slide.
All right, let me just stop and share the other screen then. I'm sorry about this. Okay, I think this
Yep.
should be working. The message is
Yes.
gone. Yeah, so I'm sorry about that. So I wear a lot of hats. I'm in computer science, but formerly part of the Global Infectious Disease Research Center, which is a COBRE here at the University of Vermont. And us being a infectious disease center made it very natural for our work to shift gear a little bit and start focusing on the COVID pandemic as early as February of last year. And our center is really multidisciplinary, covering all sides of infectious disease. So we have from vaccine testing and development to clinical work. And I'm the co-director of our mathematical and computational core. So that's really what I want to focus on today is the modeling we did for the COVID pandemic. And I'm a little ambitious. I want to cover 3 key messages. So first, the idea that epidemics are not driven by the average individual. A lot of my work is rooted in what I call network epidemiology. So we just saw a little bit the role of social networks in peer pressure mechanisms, but diseases also spread over contact networks, and social network heterogeneity plays a lot of role there, especially for COVID around the idea of super spreading events. And the fact that there's no clear rule for what makes one event dangerous or gives potential to a super spreading event. And a lot of these heterogeneities and super spreading events are driven by patterns of human mobility. So we're going to look a little bit at big data of social media data and tracking of phone to help us inform how behavior has changed over the course of the pandemic. So it's a lot to cover. I'll try and keep it big picture and give a lot of references to the work we've done over the past year. So very early on, this is an old picture now, I think from February 2020, if I remember correctly. As soon as the COVID pandemic started, at the time, sort of an emerging epidemic still, a lot of contact tracing work was taking place to try and understand the mechanisms for transmission, try and identify new cases and understand sort of the spreading potential of this pathogen. And I know you can't read the figure. It's a big figure put on a small screen right now, probably. But the idea is that every little number here is a different individual, a different COVID case, and they're not all the same. Most of them don't infect anyone, so there's not that many links of transmission events between those numbers, but some of them have way denser connections, right? So those are the super spreading events. And the idea already is that the average individual probably has no transmission, doesn't transmit much, whereas a minority of them will transmit to 10, 20, 50 people. And those are the super spreading events, and they're really important. So that's what we try and take into account in network epidemiology. And I'll try and give you, the really big picture view of what a network epidemiological model might look like. So we start with a primary case, patient 0. So that's the little node, the little individual alone on the right side of the screen. And then we might, draw a number of contacts from the distribution of contacts we know from contact tracing. So how many contacts do you have over your week of being infectious? Who are you going to infect? So in this case, my primary case infects 4 people. And then I can repeat that, ask, well, those 4 people, how many people do they infect? And I draw new numbers and I repeat again and again, and I grow a transmission tree. Now, the important thing here is that classic models, we've all heard over the last years about the SIR model of disease transmission and sort of foundational work going back to Kermack and McKendrick in the 1920s. What this work assumes is what we call a mass action mechanisms. So most individuals are the same, there's homogeneous mixing, and they all transmit to a certain number of secondary cases, and that's going to be this basic reproduction number R0. So R0 here is the average of the number of transmission, but we actually have a full distribution underlying it. And then we capture heterogeneity like super spreading events. So most of my individuals here are not infecting anyone. They're dead ends of the transmission tree. And I do have nodes that are infecting, maybe 15 people at a concert or whatever event it was. And what we want to do here is sort of repeat a lot of the classic work from epidemiological models and ask, well, what fraction of the world will get sick if it takes over the world? Or if we're lucky and the outbreak dies out, how big might it get, right? So that's what we're trying to do. And if you're curious about the mathematical detail, we have a lot of work on that, but I'd really recommend one paper that we published in the Royal Society interface on sort of going back to the work of Kermack-McKendrick and making it more general using this idea of network epidemiology. I'll just give you like a quick highlight of the type of mathematical tools that we use. So unlike Kermack and McKendrick, we're not using calculus and differential equations. We're using a tool called probability generating function, which is just a tool from the analysis of branching processes where we use polynomials to store distribution of possible events. So we use polynomials as sort of a data structure where the coefficient in front of every term of the polynomial can encapsulate the probability of a certain event. In this case, the distribution of infections that an individual might do. And it's a really useful tool. We can do a lot of powerful analysis, but most importantly, it allows us to calculate things like the probability of an epidemic and the forecasting the size that that epidemic might have over its entire course. So this is still very early on. I think in March 2020, we released our first forecast. So estimates were starting to appear for COVID-19 based on contact tracing data in Wuhan, mainly around this basic reproduction number R0, so the average number of secondary cases. So we had an idea already at the time that the original variant was around between 1.5 and 4, let's say. So that's the red box here. Then I'm showing a second parameter, which in my mind is just as important, which is this dispersion parameter, which measures how heterogeneous around R0 is the distribution of secondary case that in given case, my primary case might create. And a high dispersion parameter, it's a little counterintuitive. High dispersion parameter is actually low variance. So Kermack-McKendrick homogeneous population would be at the very top here. And at the very bottom, you have very heterogeneous outbreaks, maybe like Ebola, something more relying on super spreading events. And the key part is that in the region that we're focused on, like if your R0 is around 2, this dispersion parameter is actually super important in telling you whether you should expect an epidemic size of 10% of the population or 75% of the population. So very early on, we had this intuition that based on the range of R0 we were seeing, it wasn't so much about whether the value was 2.5 or 3.5, it was much about how much heterogeneity was going on. And very quickly, it seemed like there was quite a bit more heterogeneity than other airborne infections like the flu, and that super spreading event, so the tail end of this distribution of secondary cases, was going to be super important. So what is the dispersion parameter K? How much heterogeneity there is? That was sort of the key message at the time that we worked on for quite a few months. And COVID is sort of unique in that aspect. So on this slide, I show what the classic model, so classic SIR or SEIR model would predict as a fraction, as the potential epidemic size as a fraction of the total number of susceptible individuals initially, right? So 1 is everyone will get the disease, 0 is nobody will get it by the end of the outbreak. And we compare it based on estimates of R0 and dispersion for a range of diseases. And the key thing here is that all shades of green and blue are essentially different network models making slightly different assumptions. And in most cases, they overlap and they're not that different from the classic prediction from Kermack and McKendrick or SIR models, except for COVID, where we see like completely different estimates, whether you're assuming a homogeneous population. And we do see an outbreak like this, right? So this is a fishing vessel where 85% of people on board got infected. We do see in homogeneous settings, people do infect each other a lot. At national levels, it tends to be more like network models where the heterogeneity matters a ton. And COVID was unique in this sense. And I like to see it both as a problem. It means that it's really hard. You can't act on everyone the same. You need to figure out what are the mechanisms that makes people super spreader or that create super spreading events. But it's also an opportunity. It's an opportunity to leverage network biases in the form of interventions like contact tracing. So over the summer, when it became clear, summer 2020, when it became clear that these super spreading events were super important, we shift gear a little bit, trying to understand where are they and how can we sort of use that knowledge in our intervention. So we did a lot of work on contact tracing, and I really want to keep it big picture here and give you one key like network science intuition, if you want, about why backward contact tracing is super important. So backward contact tracing is the traditional way of doing it. It's like you have a new case, you actually interview the people, and you ask them, where were you over the last week? They were at a party. That's backward contact tracing. You identify who else was at that party, contact them, test them, right? So you go back in time, then forward in time to find new cases. A lot of digital apps for contact tracing are only forward in time. So if I tell my phone I tested positive, it's going to tell other phones that I'm now positive that they were exposed. That's forward in time. Well, the key part of backward tracing, if you take any network of transmission, and I'll focus on the panel B here, there's this like key thing in network science called the friendship paradox. So if I go backward in time and I follow a random transmission event to its root, I'm actually n times more likely to find someone who infected n people than someone who infected one. It's just a statistical bias of network. So by doing backward tracing, you identify super spreading event and you get to leverage that bias as a very powerful intervention. And we model that using the same tool of probability generating function. The key insight is that very like small level of contact tracing probability help a ton. So for any like different transmission levels, which are these different curves in the first panel, as soon as you go from zero contact tracing to a little bit, so this is the probability of tracing around a given case, you see a pretty steep decrease in epidemic probability, the probability that a cluster blows up. And so any level of contact tracing is actually super useful, especially when you have this backward mechanism, which is what the Department of Health in Vermont, for example, did a really good job with, but that new digital contact tracing app just ignored for privacy reasons. So a lot of the insights from traditional contact tracing were not implemented in most of these apps. So I'm going to switch gear a little bit here and move away from sort of these mathematical modeling, and I want to give you a bit of an intuition for why, even though it seems like we could have been able to control COVID quite well, it's always coming back, right? Why? What are these super spreading events, and what has been the big challenge over the last year? And so we did a few reviews trying to understand, what makes super spreading events. One of my favorite we published in PLOS Biology, and we studied the dynamics of that, trying to understand how localized is COVID. But really the key insight from all this work is that there is no, just the way there's no average individual, really, there's no average super spreading event. So we see super spreading event in events of all types. So this is just a scatter plot of super spreading event based on their duration. So from 30-minute at the gym to cruises for 1,000 hours, and from 10 people to thousands of people. And really attack rates are all over the place. So it's really hard because it's shaped both by the density of the event, its duration, its size, but also, are the windows open? Is it indoor or outdoor? Everything comes into play, and the context is super, super important. And so we were trying to get more data than news stories. The scatter plot I just showed was us going through all newspapers and taking notes of super spreading events. And you might have seen a lot of that. There's been quite a bit of work using human mobility. So your smartphone tracking you and essentially giving us, a digital trail of human mobility, and we can try and understand if the patterns of super spreading events are changing in time, which would make it very hard for contact tracers to reliably know what to look for. And I'll just give you one quick example to wrap up of what we find in these mobility patterns. So the data I'm showing here is from a company called SafeGraph. What SafeGraph does is foot traffic data, right? So originally, before the pandemic, it's essentially if you want to open a McDonald's, you ask them how many people walk this street corner, and that helps you pick what street corner to put your McDonald's on. But it's also useful in the context of COVID to try and understand who goes to what businesses, how many people go there, and how that patterns change over time. Now, it's not like necessarily scientific quality data, so you need to do a lot of normalization within a given location and across time, so it's a little tricky. But what we look at in this case is specifically around churches, is we can look at how number of visits change over time. So this is before April 2020 and after. And in most places across the US, you saw a significant decrease in visits to churches over time. So between 33% to 75% in some states. Vermont is the outlier. The number of visit to churches is already really low. So it's an outlier in all of this. But that's great. People are going less to gatherings, and that's what we wanted in April 2020. But then in the second panel, if you look at, well, the people that still go to a gathering at a church, what is the percent change in the distance travel? And there you see a lot more heterogeneity. You see people that travel actually more to go to a church than they did before. To us, that's a mechanism of the adaptation, right? So if your local church is closed, maybe you go 2 towns over to a church that's still open, right? And that's a problem with the fact that a lot of the interventions were non-uniform in space across different towns, counties, and states. So we see a lot of that. And we see that, I don't want to single out churches. Churches were also, the place where food banks were happening. There was a lot of reason to go to a church. And we see the same thing in grocery stores and in gyms. And in fact, if we look at what fraction of churches saw an increase in visitors or what fraction of grocery stores saw an increase in visitors, we see a slight correlation. But across the board, it's about 1/3 to 1/4 of location actually saw more business and more people during the pandemic than before. We think because of these non-uniform intervention. And that's sort of a problem. So the intervention worked globally. It decreased the connectivity of the network, right? So globally, we saw a decrease in connectivity, decrease in R0, but locally around specific places, we might have seen an increase. And that's sort of a problem where you get more and more super spreading events, or you rely, or COVID, I should say, relies more and more on those super spreading events. So I'll wrap it here. Like it's a lot, and I tried keeping it big picture, but really I think what we need to realize in mathematical modeling is that epidemics are heterogeneous and stochastic. The old models were great for intuition, but they don't really help in terms of forecasting. What do work in the old-fashioned work is interventions like contact tracing, and it's a shame that a lot of the digital work, the way contact tracing was implemented, especially in Europe, didn't quite implement those old school insights that we have from epidemiology and network science. And then part of the problem is also that behaviors change in time. So our interventions and our models have to keep changing. So the last reference I'll give is a new review that we have out in Nature Human Behavior, which is essentially what needs to be done in the future so that next pandemic we have better models to do so. And so I think my time is already up. So I'll just thank my coordinators, my collaborators, and the Global Infectious Disease Research Center at UVM. And thank you all.
Thank you so much, Oren. That's such a wonderful high-level view of this modeling effort. Are there questions from anyone? So, there's this national effort, MIDAS initiative, where they are bringing multiple models together in this scenario modeling hub. Are you a participant of that? And because your model, I think, potential could really complement very well with what they are trying to do there.
Yeah, so we're in touch with a lot of people part of that group. I'm a big fan of ensemble modeling, like there's no one model to rule them all, and you need as many models and as many perspectives as possible. So we're actually wrapping up, like trying to formalize this network epidemiological model as much as possible to be able to put it out there as a tool that will be as easy to use as possible for next pandemics and be part of this sort of global effort of modeling.
Mm-hmm. Yeah, sounds exciting. Yeah.
Yeah.
Any others?
I have a question. In your models there, are the edge lengths related to sequence identity between the nodes?
No, so right now, all of the data that we've used is really contact tracing. So we're trying to incorporate sequencing data or other types of data that can help complement contact tracing. But really right now, we've relied on actual contacts, either through interviews or through like mobile phone traces. Okay.
Great, thanks for a great talk, and I would encourage additional questions to continue in the chat. Thanks again. So we'll move on to our next speaker. My pleasure to introduce Dr. Fred Kohling, who is the co-director of the Genomics Shared Resource and director of the Single Cell Genomics Core here at the Dartmouth Geisel School of Medicine. Fred has been the lead on the Single Cell Genomics Core within the Center for Quantitative Biology, COBRE. And the title of his talk is Single Cell Genomic Solutions at Dartmouth and Beyond. And Fred, I'll turn it over to you.
Awesome. Thank you, Mike, and to the organizers and to you and Cathy in particular for inviting me to speak today. So I'm excited to talk about what we're doing as part of our Center for Quantitative Biology, COBRE, in the Single Cell Genomics Core. Really a core facility directed to bringing single cell genomic services to researchers here at Dartmouth and also throughout the region. So we've actually been doing single cell genomics work at Dartmouth for quite some time, since about 2017. But really, when we received the COBRE award in 2019 to formally establish a dedicated single cell genomics core is when things really took off. And this award has really given us a lot of flexibility to explore new single cell technologies and make a lot of investments in personnel and infrastructure in this space. And so the goal really of the single cell genomics core is to provide an end-to-end solution for folks that want to do single cell genomics projects. From the earliest stages of experimental design and conceiving of a project where we're there and available to consult about experimental design and come up with the appropriate technology for folks to use. Once samples actually enter the lab, we have a pretty rigorous QC process for examining these cells microscopically and checking out their viability and making sure that they're in good shape for the assay. And then we rely largely on our 10x Genomics Chromium platform to perform any of the workflows that we're doing in the core. So this instrument will encapsulate single cells into droplets and then uniquely tag the molecules from every cell so we know where they came from. And then we do sort of traditional next generation sequencing library preparation and sequencing on an Illumina platform to read out the data. The second half of our COBRE in terms of the cores is our data analytics core. And so we have a whole team dedicated to analyzing these types of data downstream, which is obviously a really critical part of making these assays accessible for folks. I should also say that the COBRE, as I mentioned, has given us a lot of flexibility to do research and development in the lab, to validate new workflows, test new technologies, and also provide pilot funds for projects that may be willing to use a novel technology or for a novel application. And this has really helped facilitate the adoption of these technologies here at Dartmouth and for folks elsewhere as well. So as I mentioned, the 10x Genomics Chromium platform is really the workhorse in our core. It enables us to do a very wide variety of single cell genomics assays. And there's a number of different molecules that we can profile at the single cell level. Everything from RNA molecules for gene expression. We can do epigenomics at the single cell level through single cell ATAC-seq to look at regions of open chromatin and DNA. And we can also look at protein expression in single cells as well. There are also a number of assays that are really targeted directly towards the immunology space. And so we have a number of projects where we're doing T cell receptor and B cell receptor sequencing of T and B cells, and also coupling that with assays that can measure specifically what antigens those cells are actually recognizing. And so sort of putting all of these together, you know, it's exciting to be able to measure any one of these molecules in a single cell, but our ability to measure multiple of these types of molecules in sort of a multi-omics approach has been really powerful and an exciting area to work in. And 10x also offers a dedicated multi-omics assay that allows us to do both RNA and chromatin accessibility in the same cells. So single cell RNA-seq is really, you know, the bread and butter of the core. And, you know, if you look at the literature, single cell RNA-seq is what most folks are doing these days. It's a fantastic assay for determining what cell types are present in your heterogeneous sample. And so for anyone that's unfamiliar, these UMAP plots here, or sometimes you'll see this represented as a t-SNE plot. Essentially, each dot here is a single cell, and dots that are close together in space are transcriptionally related, and ones that are far apart from each other are distinct. And so you end up with these clusters that essentially correspond to cell types that are present within your sample. And so a typical single cell RNA-seq experiment is, the goal is typically to understand what cell types are present and then possibly how they're changing, in response to a particular treatment or a different condition that they're exposed to. For the immunology groups that we support, we also couple the single cell RNA-seq to protein expression quite frequently. And we're able to do this through the use of these DNA tagged antibodies, which you can see on the left-hand side here. So, you can use an antibody that recognizes your surface protein of interest, and that is conjugated to a barcoded piece of DNA that tells us which antibody that corresponds to. And so, as you can see on the right-hand side here, we can simultaneously read out both the RNA and then also the surface protein expression if we stain these cells with a cocktail of these DNA barcoded antibodies. And this has been really useful, as I said, for immunologists that may be using tools like flow cytometry to typically identify their populations of interest. This gives them parity between their flow cytometry data and their RNA-seq data, having that surface protein expression, but then being able to dive down into the thousands of transcripts that they might be able to detect in a given cell. We're also using the 10x multiome assay, as I mentioned, to do both gene expression profiling and chromatin accessibility in the same cells. And this is a project that we've been working on with Lucas Salas's lab here at Dartmouth. So on the left-hand side, you can see a data set that is clustered based on its gene expression profiles. And then on the right-hand side, the same data set clustered based on its chromatin accessibility. And you see sort of a similar structure. But in this case, you now have information within each cell about their RNA expression and also the chromatin structure that's there. So that allows us to do some pretty neat things. We can find a gene that's expressed in this particular cluster up here at the top left. So it's based on its gene expression, and its chromatin profile is open, as you can see by the cells colored in red. And then if we look specifically in that cluster at this gene, we can see its chromatin accessibility profile across the different clusters. And so this is cluster 8 here is where this gene is being expressed. And we can see the regions of chromatin that are open, allowing that gene to be expressed. And of course, not surprisingly, we see a peak at the promoter region here, but we also see a number of other regions that can be, even several kb away that are responding to the expression of this gene. So this is a really powerful way to identify genes that are regions of chromatin that both positively and negatively regulate the expression of given genes and how that can change across different cell types. So really powerful tool for identifying novel regulatory elements that may in a cell type specific fashion. We've also done a little bit of work creating our own multi-omic assays, and this is work that we collaborated with Divakar Padabiraman here at Dartmouth. And he's been doing these experiments using a tool from a company called Selecta. They produce these lentiviruses that contain unique DNA barcodes within them. And so, and these barcodes are expressed so that they can be read out at the RNA level. And so he'll start off with a population of cells that he infects with this virus, such that each cell gets a unique DNA barcode integrated into it. And then he can split that population of cells and treat some of them with a drug and leave others alone. And so he has a comparison of untreated cells and then those that are responding to a particular drug treatment. And so when he does that, if you look at the gene expression profiles like you would in a standard RNA-seq experiment, we can see that, in this plot, most of the untreated cells reside in the right-hand side of the plot, and there's some heterogeneity there. And then when they're treated with drug, some of the cells respond in a small way, but sort of remain on the right-hand side of the plot, while others completely change their gene expression profile and produce this unique population that's over here on the left side of the plot. The advantage of having these DNA barcodes in here now, you can actually look and see these cells that have expanded on the left-hand side of the plot. First of all, they all have the same DNA barcode, which means they originated from most likely the same original cell. And then you can also look and see where that barcode is present in the untreated population before it was exposed to drug. And so we can almost go back in time and say, what was the gene expression profile of a cell that became resistant and shifted its gene expression profile and expanded when treated with the drug? We can also find anecdotes here where this population is expanded a bit after drug treatment, but its gene expression profile did not change significantly. And so this allows us to start to answer questions about whether or not cells are pre-programmed to be resistant to drug treatment, or if they're seeing the drug and changing their gene expression profile, and that's why they're able to escape. They're able to escape the effects of the drug. And so this barcoding technology is really quite powerful and allows us to do these sort of studies where we can look at before and after treatment in the same clonal population of cells. So you've probably gathered that we're pretty excited about our 10x genomics instrument. It enables us to do a number of different assays and makes it quite accessible for folks. But there are some limitations and challenges to using the platform. The first is logistics. Really the ideal sample for the 10x platform is a fresh sample, freshly dissociated from the tissue or from the cell culture which it comes from. And then it needs to be loaded onto the instrument fresh. There are protocols out there for either cryopreserving or sort of lightly fixing these samples, but there's always, the potential worry that the sensitivity of the assay will be impacted or that certain cell types will be lost. So fresh samples are best. It's also a fairly expensive assay, about $3,000 per sample to measure about 10,000 cells. And the sensitivity of the assay has certainly improved over time, but because you're measuring so many cells, you don't necessarily get a lot of depth of information per cell. And we also have 3' or 5' end bias, depending on which assay we're using. So we're really just doing gene counting to measure what's expressed, and we're not able to do any sort of isoform level detection here. So if you're interested in alternative splicing at the single cell level, this isn't really the technology for you. So we're always keeping an eye on new technologies that are out there. We're really excited about a newcomer to the field called Parse Bioscience. They've commercialized a technology that was originally published as SplitSeq. And basically the idea with this technology is that your cells can be fixed and permeabilized as soon as you generate your single cell suspension. And then they can be put in the -80 in the freezer for up to 6 months, possibly even longer. And you can accumulate multiple samples over time. So if you have a longitudinal study or just want to accumulate some samples and then just run the assay all at once, you can do that. This also will allow us to scale. The assay itself is very scalable, and the fact that we can collect samples from multiple users to do one large run brings down the per sample cost pretty significantly. One thing that we're in the process of testing now through some pilot experiments is whether this assay is actually more sensitive on a per cell gene detection level than the 10x assay, and whether or not we're able to detect specific splice isoforms, which is theoretically possible with the assay. I think the part about this that I'm most excited about is that it's going to let us collect samples from distant sites. Currently, folks can either preserve a piece of tissue or cell suspension and ship it to us, or they can drive to us to bring us a fresh sample. Both of those aren't the easiest thing to do. So we're hoping that by being able to fix and freeze your samples and ship them out, we're going to be able to help a lot of folks like yourself that might want to do single cell work, but don't have a core nearby to submit your samples to. And so we're really hoping that this allows us to branch out a lot beyond Dartmouth and more regionally or even nationally to be a resource for more folks. I want to switch gears briefly and just mention what we're doing in the spatial transcriptomics space. So spatial transcriptomics is a really exciting technology. It takes the idea of single cell analysis, where we're looking at gene expression in individual cells, and puts that information back into the spatial context, the biological context of the samples and the tissues from where these cells actually come from. And so we're sort of, merging sort of old school, histological data with this next generation single cell data into a combined spatial high dimensional omics platform. And to do this, we're leveraging 10x's Visium technology, though we're looking at others. The idea here is that you have a glass slide with these capture areas that are shown as boxes. And on spotted across this slide is a lawn of oligos that contain a spatial barcode sequence. So each spot has a collection of oligos, and each spot has a unique spatial barcode. So we know where on the slide that barcode's coming from. And those squares there are about 6.5 millimeters square. And so by collaborating with our pathology shared resource, our users are able to take their tissue section, freeze it in OCT compound and put it in the -80. They go to the pathology shared resource where sections can be cut and placed onto these Visium slides. And then in our lab, we have an automated microscopy platform so that we can image the tissue. And then we permeabilize the tissue so that the RNAs can flow out of the tissue section and onto the slide where they're captured by those spatially barcoded oligos. And then we do some molecular biology magic to capture those molecules and convert them into sequenceable fragments that can go onto the Illumina platform. And as you can see in the middle here, similar to the picture I showed earlier, this brain section, what you end up with is a picture of the tissue, but with all of the gene expression profiles overlaid on them. You can search for your favorite genes and find them within the tissue, or you can agnostically cluster the data and identify regions of the tissue that have specific gene expression patterns. And so we're really excited about this technology as sort of a bridge into the clinical space, really doing true translational research. The single cell genomic assays on the Chromium platform are very discovery based. Being able to put that into the context of the tissue gives us parity with a lot of the clinical data that's out there. Where the Visium assay is not targeted, it captures all polyadenylated mRNAs, and so it's still a discovery-based platform, but it's in the context of tissues. And this provides a nice complement to some of the assays that our pathology shared resource are looking at, spatial assays that are targeted. So they may have a panel of genes that they know are important. They want to see where they're spatially located in the tissue. And so Visium provides a nice complement to that, where we can help them identify what markers are going to be useful for their targeted spatial assays. And I think this is going to be a really rich partnership between our group and theirs. The COBRE has really given us the ability to expand our infrastructure, as I mentioned, and this includes our sequencing capacity as well. We recently received an S10 award from the NIH to buy a new NextSeq 2000 sequencer, which is going to more than double our sequencing capacity here, which is really essential for these single cell assays, which take a lot of time and money and effort to sequence. So our throughput's going to go up considerably. And the pathology lab downstairs also just purchased a NovaSeq instrument. So when we have projects that are considerably large or expensive or need a faster turnaround time, we'll have the ability to leverage that instrument as well to bring our costs down, bring our turnaround times down. And again, with the goal of making this more accessible to everybody. Once the data comes off of the sequencer, I mentioned that we're integrated with the data analytics core, which is the other part of our COBRE. And they do all types of data sets. They do standard bulk RNA-seq and ATAC-seq data sets, ChIP-seq, et cetera. But they also do have expertise in the single cell space as well. They will initially take a single cell data set and run some pre-processing on it so that you have sort of take your raw sequencing data and generate a gene expression matrix that's amenable for downstream analysis if that's something that you want to do on your own. But if you have a more complicated project, possibly with a more complicated experimental design, they can be incredibly helpful at wrangling those types of data sets. And so one thing that's really important in single cell data is controlling for batch effects and variation across individuals or different treatment groups. It's really important to tease out the batch effects that might be the result of sample collection versus the actual biological variation that's there. And so they use a a number of different methods, but one of them is the data integration procedure that's available in Seurat, which is an R package that's used for single cell analysis. And this allows you to integrate data from different sources and to sort of normalize out sources of technical variation. And so a project that they had conducted with Matt Haverda's lab here at Dartmouth, looking at Parkinson's disease patients. You can see control samples on the left and Parkinson's on the right. And before you integrate the data sets, you see this sort of batch effect, just from one patient to another, one collection day to another. After you run the integration procedure, you've normalized out those batch effects or patient-specific effects, and now you can actually do your comparisons more accurately. Another important goal of single cell analysis is to understand what cell types are actually present in your sample. A lot of times this is just done by looking at your favorite genes and deciding which clusters those genes are expressed in and saying that is your cell type of interest. But the better way to do this, especially as more large-scale single-cell reference data sets are coming out, is to take your data set and map it onto some high-quality, well-established reference data set. Basically the single-cell equivalent of mapping to the human to the human reference genome. And so that's exactly what we're showing in this slide here. The data analytics core does this fairly frequently, where they're taking a data set, an RNA-seq data set, and mapping this onto a larger reference data set to identify the cell types that are in that sample. This method can also be used to map different types of data onto each other. So if you have a 10x genomics Chromium data set and there's a reference out there that is SMART-seq2 or DROP-seq or single cell from another technology, those can be integrated as well. But it's a fairly complicated process. And again, that's something that these guys are really well versed in. And you can just see here the labels being automatically applied here to this Parkinson's data set, sort of agnostic of any user input. So with that, I want to acknowledge everyone that makes everything happen in the single cell core. Mike and I are the co-PIs. We really just send a lot of emails, I think. Heidi, Carol, and Liz, who actually just left us for a job with a single cell company in Boston, really do all the heavy lifting in the lab. On the data analytics core side, Dr. James O'Malley is the director there, and Owen, Shannon, and Tim do all the heavy lifting, and they're fantastic to work with. So with that, I'm happy to take any questions and hope we have a chance to work with some of you guys in the future.
Thanks, Fred. I think we're running behind schedule, so we may have time for one quick question. I'm going to ask the rest of the questions, maybe go to the chat. Okay. So to keep things moving along, please put your questions to Fred directly in the chat, and Fred, you can hopefully answer them as we go along here in the session.
Sure.
Thank you very much for a very nice talk. It's my pleasure to introduce our next speaker. Dr. Ritambra Singh is a project leader in the COBRE Center for Computational Biology of Human Disease at Brown University. She's an assistant professor of computer science at Brown University. And the title of her talk is Integrating Long-Range Regulatory Interactions to Predict Gene Expression Using Graph Convolutional Networks. So Dr. Singh, welcome, and we'll let you take it away.
Thank you so much for the introduction. So good afternoon, everyone. My name is Ritambra, and I'm an assistant professor at Brown. And today I'm going to talk about a very exciting project in the lab where we are trying to integrate the 3D spatial information of the DNA when modeling gene expression. So starting from the very basic, we all know DNA is the basic building blocks of most organisms, and the DNA gets converted to RNAs, which gets converted to proteins, and proteins are the structural components of the cells, and the cells, they make up the organs. And the first three steps are very important for researchers to study because a lot can go wrong in these early steps, which can propagate further down the line and cause all sorts of diseases. And the reason a lot can go wrong is because the genes that eventually get transcribed into proteins are regulated by a very complex machinery. So to give you an example, transcription factor binding protein has to come and bind close to the gene to start the transcription process, which is the conversion into the proteins. This in itself is related to whether the gene region is closed or whether it's kind of open. So the DNA is kind of wrapped around these proteins called histones, and these histones have tails that are prone to chemical modifications known as histone modifications. So the idea is that if this chemical modification is repulsive in nature, it will push and kind of cause the region to open. And if the chemical modification is more attractive in nature, then it would close the region by attracting the histones. And so these relationships are important because they eventually control gene regulation. And one important question to try and understand is how are these histone modifications related to the gene expression? And the reason we want to do that is because these changes are potentially reversible. And so there is a lot of promise in developing these epigenetic drugs that could potentially control the gene expression. So in order to try and understand these relationships, we have these amazing technologies with which we can measure the gene expression in different cell types. And we can also measure the different histone modifications and figure out where they are taking place and what's the signal strength. But the issue here is that it's a lot of data, and we are looking at approximately 20,000 genes. There can be multiple combinations of around 20 histone modifications, and we are dealing with hundreds of cell lines. So if an analyst were to start making these changes and seeing what could be happening, this task is almost impossible to do. So back in 2016, we came up with this idea of why not use deep learning models to try and learn these combinations of histone modifications. And so the idea behind trying to do this is because deep learning models have this very wonderful property of automatically extracting all the relevant features in trying to make accurate predictions. So to give an example over here, for this particular deep learning model to predict this image as a public park, it will not only capture the local features of the image, but also capture the combinations of these local features or the global features to make the accurate prediction while it's trying to train from the data. And so our idea was to use the histone modification signals as the input and connect it to the gene expression as the prediction that we wanted to make. And the idea was that these regions, subregions from where we were collecting the histone modification signals could be the local features, and the combinations of these histone modifications could be the global features. And so a deep learning model made sense for this particular concept and idea. So to our joy, we found that the model was able to do very good predictions of gene expression. And so we could claim that it could capture the combinatorial relationships of the histone modifications. But more importantly, we did what is known as interpretation in deep learning literature, where we were able to highlight what features were relevant for the model to make a particular prediction. So in this particular example that I'm showing, the model was able to say, was basically focusing on H3K27ME3 and H3K9ME3 marks to predict that the gene was off. And we found in the literature that these two marks are biologically known as repressor marks. And so they are basically involved in the repression of the gene expression. So this was something that the model picked up from the data, and it was very exciting. Now, one thing that our model did not do at that time was to capture the long-range interactions when it was modeling this gene expression. And this is not only our work, but most of the recent works is also are not including this long-range interaction information. So this brings me to complicating the picture even further Because so far I've been talking about these local features, these histone modifications controlling the gene expression, but now I'm bringing into the picture the way the DNA is arranged in the 3D space. So you can think of a histone modification, which is actually quite far from the gene if we were to just take the DNA and make it linear, but in the actual 3D space, it's looping and is very close to the gene, and it could affect the gene expression and cause diseases. And this is not just a hypothesis. There are evidences in the literature where these long-range interactions have been known to cause disruptions and cause diseases. So keeping all of this in mind, our aim for the project is to try and integrate the spatial information to the existing framework that we have, where we are taking the histone modifications as the input and predicting the gene expression. So the first idea is to be able to just do that prediction task while incorporating this information. But more importantly, just like our previous work, we still want to do some interpretation, that is, figure out what features are relevant for the model to make a certain prediction. And this is important because then we can kind of compare these set of features between a healthy cell state and a diseased cell state. So the information that we are using is the DNA organization. And the reason that this has not been incorporated is because until very recently, we were not able to measure this information. But now we can, using these experiments called Hi-C experiments. So the output of the Hi-C experiment gives you this matrix that captures the 3D conformation of the DNA. So the rows and the columns of this matrix are the different regions of the genome, and the value of this matrix represents how frequently those two regions are interacting. So a high value means that those two regions interact very frequently, and a low value means that those two regions don't interact. And so our idea in this is to take this matrix and convert it into graph. So in this graph, the nodes would represent the genomic regions, and we connect two nodes if those two regions are interacting based on the Hi-C data. And if they're not interacting, then there is no edge between them. Now, before I go any further, I would like to mention that this project is being led by my PhD student, Jeremy, and two talented undergraduate students in the lab, Zavi and Shalin. Right. So now once we have formulated the Hi-C data as a graph, we can apply what is known as a graph convolutional neural network. Now, you may be familiar somewhat with the regular convolution on images, where to learn a representation of a pixel, when we are performing this convolution operation, we take the weighted aggregation of the neighboring pixels. Similarly, in graph convolution framework, to learn the representation of a particular node, we take the weighted aggregations of the features of all the neighboring nodes. And this feature could be any numerical number capturing some property of that particular node. So the input into a graph convolutional neural network are the features of the node and an adjacency matrix used to represent the graph, because that is what gives us the information of what are the neighbors of the node on which we are doing this operation. So this brings us to the formulation of graph convolution neural network. There is a lot of information here, so let me walk you through it one by one. So given a particular graph, suppose we are doing this operation on the node with the star on it. We start with its original node features that we have collected from the data. And then, as I mentioned, we take the weighted aggregation of the features of its neighboring nodes. And here you can see we are also normalizing by the size of the neighborhood. Now, this step can vary from different formulations to formulations, but we include it where we kind of also add the output of the previous convolution operation. So in this case, we haven't done it yet, so it would just be the original node feature. So we add that information as well. And then finally, we have a non-linear function, which are very common in deep learning models. So that's one round. Now, this gives us the convolution output for layer 1. Now in layer 2, we will repeat this step. And then after k times, we will have this final representation of the node, which is capturing the information from its neighborhood. And then we can use this for any downstream prediction task. So how does it all fit into our framework? Given a gene Y, suppose, we have its gene expression, which is the output, the quantity we want to predict. And then for this particular gene Y, we look at its genomic region and we find out what are its neighboring genomic regions that it's interacting with using the Hi-C map. So we have the list of its neighbors. And then as far as node features are concerned for all of these different genomic regions, we take the histone modification chip-seq signals, and that's the numerical quantity that ends up becoming our node feature. So we have the node features and we have the information of the neighboring nodes. So this fits very beautifully into our graph convolutional neural network framework. And once we have learned the representation of that particular node, which is representing gene Y, we feed it through some fully connected layers and we do the final prediction. Now, as the model sees these samples again and again, it's trying to reduce the error between what it's predicting and what the true value is. And in this way, it learns meaningful relationships. So for our experimental setup, we ran it on 3 different cell lines. And as I've already mentioned, the input is the graph structure generated from the Hi-C map. And the node features are these 6 different histone modifications that we chose as the feature, as the numerical features for each node. And the output is the gene expression signal. So first thing first, prediction results. We find that after we have trained the model does a reasonable job of predicting the gene expression values, and we see high correlations between predicted and observed values. We also compare it to some existing deep learning baselines, and overall we find that the model is doing a good job compared to the baselines when it comes to prediction. But as I already mentioned, this is just telling us that the model is capturing something reasonable from the data and it's capturing some relationships from the data. But what is more important to us is how can we extract this information that the model is learning. And so to do that, we apply this method called GNN explainer. And what that does is, on a high level, is given a particular node, it assigns contribution scores to the node. So it tells us what nodes are important for the prediction of that particular node. And then we are also able to get these important scores for the features. So what histone modifications are relevant for the prediction of that particular node. And the way we do that is by minimizing this objective function. And so what is happening here is we are optimizing the fractional masks M and N. So M and N are just masks that we apply on the graph as well as on the node features. And what we are trying to minimize is what is the original prediction of the model, and the error that the prediction of the model gives once we have masked certain parts of the graph and certain features. So this is conditioned on the fractional subgraph mask and the fractional feature mask. So you can think about it, if we mask certain nodes out and let the information pass and the prediction error is high, that means that that node was important, right? And if we mask something and the model makes reasonable prediction either ways, then clearly that node was not important. And this is the same thing for the features. So when we do that, we ran it on a couple of a number of genes, and here I'm just showing you example of 4 genes in K562 cell line, which is a leukemia cell line. And so here what I'm showing here is the yellow node is the node that is being predicted, and it's surrounded by all its neighboring nodes. The nodes size of the node is representing the important scores that GNN explainer gave, and the red is representing the actual evidence of interacting nodes that we got from the study listed below. So what we can see is that for the actual nodes that are interacting with that particular node and affecting its gene expression, those are being assigned high scores by our model. So again, this is something that the model is learning from the data. Now, there is no way for us to validate this, but we can also get these important scores for the histone modification signals. But one thing to notice is that the 2 signals that we keep getting important scores for are H3K27AC, which is an enhancer mark, and H3K4ME3, which is a promoter mark. And in all the recent studies which are trying to learn about these long-range interactions, these 2 marks are used very frequently. So it's kind of comforting that the model is picking these 2 from the 6 histone modifications that we have chosen. So to summarize, we can say that GC-Merge, our tool, is able to integrate the spatial information to model gene expression. It's able to capture the long-range interactions, but more importantly, we can get these explanations from the model that can serve as hypothesis-driving tool. And we have the preprint and the code available if you're interested. And finally, I would like to thank all the lab members of my lab, seen here in a socially distanced lab hangout, and especially Jeremy, Shal, and Zavi, our collaborators, Erica, and of course, this research was funded by COBRE. So thanks to all the members of COBRE CBHD at Brown, as well as members of all my affiliations. Thank you, and I'm happy to take questions now.
Thank you for a very nice talk. We can take one quick question, and then I would encourage people to put additional questions in the chat. Is there someone that would like to unmute themselves and ask one? So to keep us on time, we'll move questions to the chat, but thank you very much. That was a really nice talk and really nice work.
Thanks. Yeah, thank you. Thank you for all the speakers. It has been great. So our final speaker of the session is Dr. Claudine Djokovic, who is Director of Clinical Research Institute for Researching Equity and Community Health at Christiana Care. And she is also lead of the Biostatistics, Epidemiology and Research Design Core of Delaware ACCEL, CTR, as well as Director for Center Research Support Network at Delaware INBRE. Unfortunately, she won't be able to join in person since she's traveling on a plane actually right at this moment. But we will play her pre-recorded video. So Lauren.
Good afternoon, and thank you for attending this presentation. I would like first to thank Dr. Wu and Dr. Whitfield and the Rhode Island IDEA program for giving me the opportunity to present our network story, and do apologize for not being virtually live. Here is what we'll discuss in the next 15 minutes. I will first give a brief review of the IDEA program in Delaware, then describe the INBRE CSR core and the ACCEL CTR BERD core, then provide some information about the N3C cohort, and finish by describing a few examples of research projects highlighting our collaboration. As Dr. Stanhope, the INBRE PI, always says, it is all about the network. In Delaware, we have the 3 IDEA programs, INBRE, CTR, and several COBREs across our institution. Services are shared between the IDEA programs, and Delaware investigators benefit from all these services. The 3 programs fund pilot projects, INBRE provide core access awards, investigators have access to biostatistics, epidemiology, and research design through the CTR BERD core, and to community engaged research through the CTR Community Engage and Outreach core. Finally, both INBRE and CTR have a professional development core. Delaware, INBRE, and ACCEL CTR institutions are very similar. INBRE and ACCEL CTR both comprise University of Delaware, Nemours, Delaware State University, and Christiana Care. Delaware Technical Community College is an INBRE partner, whereas MUSC is an ACCEL CTR partner. Finally, Wilmington University, Wilmington VA Medical Center, and Delaware Bio are affiliated institutions to INBRE. This is a very simplified scheme of the INBRE structure. INBRE includes several cores in addition to the administrative core. A very active bioinformatics core, the centralized shared resource core or CSR, and the education and professional development core. The CSR includes 2 subcores, the centralized research instrumentation core or CRIC and the centralized research support network or CRISN. I will now focus on the centralized shared resource core or CSR. The CSR has 4 specific aims. I will not go through each of those, but again, it is all about the network. Expand, strengthen, enhance, and develop. As I mentioned, the centralized shared resource core is made of 2 subcores, the CRIC, which includes multiple instrumentation core, and the CRISN. Partner cores in the CRIC are funded by the INBRE program. Their staff have funded effort, whereas the affiliated cores are not funded through the INBRE program. The affiliated core receive core access awards. For example, a Delaware investigator might have a research project for which histochemistry analysis is needed. The investigator will discuss the cost with the core director and then will submit an application for a core access award of up to $4,000 to fund the analysis. The Research Support Network supports access to data. Core access awards are provided to investigators who need claims data for their research projects, whether Medicaid claims or claims from the all-payer-based database. Finally, all investigators from INBRE institutions that are also CTR institutions can request biostatistical support from the BERD, which is both an ACCEL CTR funded core and an affiliated INBRE core. This is again a very simplified scheme of the CTR structure. I will focus only on the BERD. The BERD has also 4 specific aims. First, to provide consultative service, a very active consultative services. And I'd like to emphasize the 4th one, which is to leverage the deep biomedical informatics and bioinformatics resources. Contrary to many CTRs across the country, the ACCEL CTR does not have per se a bioinformatics core because INBRE has a very active and very important bioinformatics core. So over the years, we have developed strong collaboration with our INBRE colleagues at University of Delaware to leverage these bioinformatics resources. As shown here, scientific staff from all CTR institutions participate in the BERD. BERD is composed of biostatisticians, epidemiologists, data analysts, bioinformatics scientists, computer scientists, and medical informaticists. Submitting a BERD request is simple. First, the investigator needs to go to the Delaware CTR website. Then they click on this box, submit a BERD consult. Then they will need to indicate what is the focus of the projects. Could be violence prevention, preventive care, COVID-19. And then indicate which type of help they need, which could be study design or methodology, biostatistics or data analysis, data analytical plan for grant proposal. And also, we've just added help to access the National COVID Cohort Registry and Data Enclave, which is a perfect segue for my next slide. So the National COVID Cohort Collaborative, or N3C, is a huge database, which as of July 27th of this year included more than 6.4 million patients. Of those, 2.1 million patients who have COVID, and more than 200 projects are now accepted by the N3C. N3C is a partnership between NCATS supported CTSA hubs, the National Center for Data to Health, NIGMS, which is very important for INBRE and CTR because it's a NIH division that supports INBRE and CTRs, and it's all overall supervised by NCATS. So for some more information about N3C, data transfer agreements are now executed with more than 80 hospitals and healthcare systems. Institutions transfer electronic health records relevant to COVID-19 to the N3C data enclave on a recurrent basis, in general every couple of weeks. The data transferred includes the records of patients who were tested for COVID, whether they are positive or negative, and for also patients who are symptomatic for COVID. All the EHR must be transformed into a common data model prior to being uploaded on the N3C enclave. So that's for the institutions to transform their EHR. And then the N3C data harmonization team ingests the dataset, runs quality checks, and transforms different data models into a harmonized analytics dataset. Very important, the N3C data enclave is a secure cloud-based environment. All work must be done on the data enclave and data cannot be downloaded. And I would strongly encourage all of you to check this website, the N3C website, where there are lots of information about the data and very interesting studies. So there are 9 CTRs around the country that provide data to N3C. And for us, again, it's all about the network. In Delaware, the team that provides data in N3C is the NEMORS team, our colleagues from NEMORS. They provide data again every 2 weeks from their patients to N3C. And then BERD scientists help investigators to access N3C through BERD request, as we have already seen. The access requirements to N3C vary by data level. The easiest requirements are for a synthetic dataset. So this is interesting because the data are derived from real data from the limited dataset that resemble patient information, but are not actually patient data. Highlighted in red are the requirements that are common to the 3 levels. Investigators need to have N3C registration, an N3C data enclave account, a data use agreement executed with NCATS, NIH training completion, and an approved data use request. The next level is for de-identified dataset. Some research projects need de-identified dataset. In this dataset, the patient data are the true data, but dates of service are shifted to protect patient privacy, and patient zip codes are truncated to the first 3 digits. Again, the common requirements are highlighted in red, but in addition, investigators need to have human subject research protection training. And finally, the next requirement is for a limited data set. Here, again, the patient data are present in this data set, but also dates of service and patient zip code. The eligibility is restricted to researchers from United States-based institutions. And in addition to the common requirements, again, investigators need to train for human subject research protection and also need to submit an IRB protocol to their local IRB. The NC3 data elements include demographics, symptoms, lab test results, procedures, medications, medical conditions, and physical measurements. To work with NC3, you need to know Python or R or SQL or Atlas. There are more than 200 projects right now in the NC3 data enclave, and all the projects are listed on the website. It's a very transparent process, and the NC3 leadership really emphasize team science and collaboration. I've listed only 4 of these projects here. A paper was recently published describing the N3C cohort, and it was published in JAMA Network Open. And finally, I listed again the websites, which I really encourage you to consult. Finally, I would like to highlight research projects that illustrate the strong collaboration between INBRE and CTR. Dr. Jingjin from NEMORS received INBRE pilot funding for her project related to ophthalmology sickle cell disease in children. But prior her submission, she was helped by BERD statisticians who helped her designing her statistical analytical plan. Likewise, Stephanie Gorino from NEMORS also received funding for her ACCEL CTR pilot related to improving chronic pain outcomes in adults with sickle cell disease. And in addition, she just was awarded a Core Center Access Award to study healthcare utilization in Medicaid patients with sickle cell disease in Delaware. Finally, Cathy Fitzpatrick from University of Delaware received also funding for her pilot related to long-acting reversible contraception. And she was also just awarded a Core Center Access Awards to study how Medicaid policies affect use of primary care. This is the end of my presentation. If you have any questions, please do not hesitate to email me at this email address. And also, I'm sure that there are some BERD members in the audience, as well as Dr. Wu here, who probably could answer a lot of your questions. Thank you again.
Thank you. So that concludes the final presentation. I don't know whether there are specific questions people would like to raise regarding Claudine's presentation, but I think this is, we have about 5 minutes left. This would also be a great opportunity for us to have more conversation. So overall, this is really a great session attended by more than 50 participants. And we have already seen very rich discussion topics in computational biology, data science, and epidemiology, covering research, covering training programs, covering core facilities. So as an outcome and deliverable from this session, in the remaining few minutes, maybe we can continue our conversations to see what kind of activities that we might want to be engaged in across institutions to foster some connection of our resources, leverage, the different capabilities and capacities that we have collectively. So it's open for everyone.
One thing that's come to mind for me, I think we've even discussed this, among my own COBRE, is trying to do our best to advertise some of the sessions and educational sessions in particular that occur across different IDEA programs across our states, and how much could we leverage that to increase our impact. Obviously, I think there's questions of numbers of people that can be involved in any one of those programs, and maybe that's the one of the limiters. But it seems that there could be a priority list by which you open it up to your local group, then to the broader group as time goes on.
Mm-hmm. Yeah, Kelly, I don't know whether you wanted to share the experience of the NEBC. Well, for this, we didn't have the core bioinformatics collaborative meeting because it's virtual. But in the past, we always have this kind of gathering, right, in the NARIC meeting. And it is an excellent mechanism. I mean, actually a collaborative that would have, a lot of continuing activities throughout the year, and not limited to just a one-time, conference and symposium.
Yeah, actually, Cathy, we Chris got us together. When was that, Chris? Monday? Monday after the meeting.
It was so long ago in my mind.
It seems like it. And we just to have a chat, and it was good. And we did, Mike, one of the things that we typically talk about are what sort of training opportunities we're providing, and I think it's a great idea to somehow have a common place or share those amongst our programs better. I mean, we do, but a lot of it is word of mouth and not as timely as it could be. The other thing that we did come up with was one that we've been working on, and I know everybody kind of has to work on, and that is training onto the cloud for, particularly for our those of us that have INBRE responsibilities and supporting research, at PUIs, we've been working on trying to come up with a way of having that other modality for conducting bioinformatic analysis. And so that's one of the things that we talked about in the small group that could get together on Monday.
Are there questions or comments or discussion points? Questions for our speakers who we didn't get a chance to ask questions to directly?
Well, I didn't get to respond to Brett. Brett asked a question about what's going to happen. Essentially, it boils down to what's going to happen, what kind of testing and what that's going to look like, and what are the best practices, especially at universities in that kind of environment. And and he also mentioned that this Delta variant is kind of a game changer. I really have no idea, to be honest with you. It's a situation where everything is different than it was last fall, in the sense that no one was vaccinated last fall, essentially, and the variants were not behaving this way. And we are continuing to test and will respond, but I suspect in the next 3 weeks we're going to know quite a bit. And one of the most insidious aspects of this Delta variant is that it's changed. It's come on the scene so fast that a lot of there's just things we don't know yet.
Yeah, I mean, I can actually add, I guess, one thing to that is that at least among the Dartmouth testing, which has now gone to reduced, it's much reduced now, often they're testing. There's now a great increase in numbers of cases among fully vaccinated individuals. And I think we're at a 93 or 95% vaccination rate among our campus, but we're seeing breakthrough infections among vaccinated individuals.
Yeah, it'll definitely be interesting. Thanks for getting back to my question. I asked it a long time ago, but I think it's on all of our minds that we just don't know exactly how we're going to handle things moving forward, both in and out of the classroom. So thank you for getting back.
Yeah, I guess I would just, building on what I talked about in my talk, that I would say that as we've been talking about moving forward, it's keeping contingency plans in place so that we can smoothly move back and forth. Ideally, I'm hoping I'll have people walking in the door at the first week of November, but it's going to depend upon what happens progressive between then. So that's why this, both for educational reasons, but also just very practical healthcare related regions, we're going to continue forward with this idea of trying to combine having both virtual and in person available for almost anything and everything we're setting up.
I mean, that's interesting to hear. I mean, I know there are plenty of institutions who have promised a fully in-person experience for the fall, and I'm not really sure they can necessarily promise that. I think part of the challenge for this from a policy-making standpoint at the college and university level is how do you make responsible policy that is also supportive of the students? Right, I have colleagues who they're in the never again Zoom category, and then because it was just they're traumatized in a way, and I understand that.
Yeah.
But looking at the fall, I think we all sort of have to be ready to pivot, and we have to think about how changing policies with regard to how we handle individuals who are in isolation or quarantine, whether they're vaccinated or not, will impact the classroom experience for our students. So definitely a challenging landscape.
Yeah, I guess I would follow that with, you know, a very realistic statement that I live in a very different world than most of you do. You know, we are a small institution. We interact with and bring students in here. We bring work participants in, but we don't have. I mean, we were in many ways fortunate. We're such a low density place that we only had about 6 weeks of time really away from our building. We're so low numbers, and it is a different world. And so when I talk about what we were doing, it's really these temporary short-term engagements with other people. I think you live in a very different perspective, different situation than that.
Definitely.
Sounds good. So Michael, do you have any final words? I think we are 4 minutes past. This has been, again, a tremendous session. Really, we all learned a lot.
Yeah, I just want to thank our speakers for doing such a great job, giving nice talks, for going with the flow in terms of answering questions, both live and on chat. And if there's additional questions, please email them directly. You can probably find them online. I'm glad we could have some discussion. I kind of miss seeing everyone in person where we can sit around the table and have discussions, usually after sessions about things like this.
Mm-hmm.
So I look forward to being able to do that with you in the future.
And next NERIC is going to be in Delaware. So we hope to welcome you to beautiful Newark, Delaware. Until then.
Sounds great. Thank you, everyone.
Thank you, everybody, for attending. And thanks for making this such a great NERIC. For those of you who don't know me, I'm the program coordinator for Rhode Island INBRE. And I have been really impressed across the board with how these sessions have worked and how everybody has really made the virtual environment work. So thank you so much. We will be distributing a survey to you guys probably this week. So please stay tuned. And thank you so much. We appreciate all the attendance and all the great presentations.